Hi everyone, let's just wait for 2-3 minutes and then we'll get started. Okay, I think we can get started. I'll just share my screen. Okay, I hope that's visible to everyone. Great. All right, so today is the final week of your coursework and this is the final problem solving session as well. So I think the last week of coursework was a little more sort of less specialized and a little kind of exploring more topics that were not covered earlier, like, plant cell mitosis, neurons, and an interesting pathway in which the Allen Cell Institute have set up to study sort of genetically labeled different components of a cell, so addressing many cell biological questions, and of course stem cells and um, what they do in the body, how they're produced, induced pluripotent stem cells. So all of that was covered, and I'm going to go through some interesting topics, and also I wanted to also talk about some analytical techniques that have been used in the research that has been shown to you in the coursework. I thought if I go into them a bit more, it might help you understand how this work was done, and of course maybe help you, in the future when you use these techniques in the lab or study about them. So that I'll also be covering. So I hope that's useful to you guys. And of course, as per usual, you should be free to ask me questions at any point. Okay, interrupt away if you need to. Okay, so the first thing that you guys learned in the coursework this time around was about plant cells and how plant cell mitosis takes place. So it's a lot like animal cells in the sense that they're both eukaryotes. They're not too different, but there are a few key differences between how plant cells undergo mitosis, and particularly at the steps of telophase and cytokinesis, we see some very distinct differences. And I'm going to cover that. So here, this is just an image showing the nuclear, or basically the DNA labeled within these cells, and you can see different phases of mitosis. As you must remember that we have initially interphase, then we have the chromosomes condensing. So you can see that during prophase, then we have metaphase where they line up, then they start separating in anaphase, separate further until they start dividing up in telophase, and then eventually the cell plate will form in between, and you'll have two completely separate cells after cytokinesis takes place. And finally, when the two daughter cells are going back to their interphase state or their G1 phase, you'll have the DNA decondensing again. So it's not in these distinct chromatids anymore, but it'll kind of become more diffuse and strand-like. So the same stuff is happening, but I mentioned something which was the cell plate. So that is something that forms during the cytokinesis step when the cells are separating. So that difference will be highlighted soon. I'll show you a video about that hopefully will clear up any questions. This is another photo I got, which is of mitosis in a plant root, and this is a confocal micrograph. The previous one was not a fluorescent microscopy. Here they have labeled the DNA or the chromatin in green, and the cell walls have been labeled in a red fluorescent protein of some kind. Yeah, that you can see the different phases of mitosis here as well, not as clearly as in the previous one, but you can tell over here that the DNA is separating into the daughter cells. Here it's sort of condensing. So different phases in different root cells. Okay. So this is a video that I found that helps understand the difference in the cytokinesis step in animal cells versus plant cells. So please let me know if you can't hear it. If you can, then all good.
In cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
So that's a simple explanation of what's going on, right? So in the case of your animal cell, you have them sort of drawing together. So you have an outward to inward movement that eventually divides these cells, right? With a ring of microfilaments that are contractile in nature. And in the plant cell, you have these vesicles that start depositing here, and you have more of an inner to outer movement where the cell plate grows from here outwards. Yeah. So I hope that's clear.
Cells.
Oh, okay. And I found one more video that unfortunately I couldn't add here. So let me just show it to you guys on my computer. So this is the one.
Plants grow, like all living things, by enlarging and dividing their cells. But plant cell division is unique in that during the final stage, called cytokinesis, a structure called the cell plate is formed that transforms into a new cell wall. Cell plate assembly occurs through the cooperation of cytokinetic vesicles and the phragmoplast, which consist of a bipolar array of microtubules and actin filaments. Golgi-derived cytokinetic vesicles, guided by the phragmoplast, accumulate at the division plane, where they fuse to form a vesicular network. A phragmoplast ring is formed by the breakdown of microtubules in the center and assembly of microtubules at the leading edge. Callose is deposited inside the cell plate as it begins to smoothen and transition to a fenestrated sheet. The continued expansion of the cell plate forms an almost continuous planar fenestrated sheet. The cell plate eventually fuses with the parental cell wall to form a cross wall. Let's look at each stage in greater detail. In the early stages of cell plate assembly, cytokinetic vesicles begin to fuse to form long tubular structures. These events are highly regulated and require the help from several proteins, including Rab GTPases, tethering complexes, snare proteins, and dynamine rings that build tubulovesicular structures. As the cell plate morphology evolves, the specialized polysaccharide callose is transiently deposited in the lumen. Callose is thought to stabilize the membrane network structure and provide a spreading force for cell plate expansion and transition to a fenestrated sheet. The cell plate undergoes morphological changes.
So a fenestrated sheet, fenestra means a hole or a puncture. So it's like a sheet with a lot of little pores, okay, in case anyone was wondering.
To reach its final shape and size, as clathrin-coated vesicles recycle excess cell plate membranes. Cell wall assembly is complete when the cell wall organizes into the typical meshwork of glycoproteins and polysaccharides, including pectin, hemicellulose, and cellulose, which replaces callose. After the remainder of the fragmoplast completes the finishing touches, the two daughter cells are finally separated by their shared cell wall.
Okay, so that's great. Right, great. So I'm just, oh, cool. So I hope that was clear. It's a little more in detail, but I thought it's useful for you to see how there's an outward movement after vesicles start depositing. And sort of creating this network of complex carbohydrates that moves outwards and eventually forms this sheet. So as we know, in the animal cells, after dividing, they sort of move apart. They may not necessarily be in the same place. They might be next to each other or they might not. But in this case, typically plant cells, they're not exactly migratory, they're not motile. So once they divide, they just partition and then they grow in size and obtain sort of like an adult size cell, which is quite different from animal cells. Another difference, which you would have seen here, I think it is there here.
Cells.
Yeah, no, it's not really there over here. So you'll notice here that the spindle fibers, they are not organized by the centrosome. So that's because plant cells don't have a centrosome. It's an animal cell thing. So they have these sort of diffuse foci that help organize these microtubules or spindle fibers for pulling apart the chromatids.
Cells.
That's another important point of difference between plant cell and animal cell cytokinesis and mitosis in general. So I have some questions here, just two, I think, before we move to the next topic. This is the first one. So which statements are true? Which statements are the true difference between plant and animal cell division? Um, yeah. So please feel free to unmute and tell me the answer, or you can message it, up to you. Hmm. Oh, I think I only made a mess up here. The right answer, I think, is not there in the options. Oh no. Just tell me what you guys think. Which of the statements are correct? I will correct the answer when I upload it. I have messed up the A to D options, I think. Anyone? Someone says 2, 1, and 4. Yeah, yeah, that's correct. Everyone's getting it right. Good job. So that is the right answer. Uh, my bad. So let's go through the options once. So the first statement is that the spindle microtubules of animal cell arise from the centrosome, while in the plant cell they arise from diffuse foci. So that's the correct thing. That's what I mentioned earlier, that plant cells don't have centrosomes. So then the next one obviously is wrong. Spindle microtubules of plant cells arise from a centrosome. That's not correct. Then the third option, the third statement is plant cells undergo cytokinesis via a contractile ring. So that's again not correct because they have a cell plate forming during the cytokinesis. So it's 1 and 4. I'm just going to change that right now. Yeah, cool. So the innermost layer of the plant cell wall is which of these? So do you guys remember? So this one's a bit counterintuitive actually. Yeah, so one person's got it right. It's A, okay? It's the secondary cell wall. It sounds like it would be something that's outside, but if you look at this diagram, right, you can see this is the cell, the cytoplasm, then the blue thing, that's your cell membrane. This is the secondary cell wall of a cell. This region here, that's the middle lamella, and this outer one, that's the primary cell wall. Okay, so this thing is the blue region after the membrane, that's the secondary cell wall. Primary cell wall, and then you have the middle lamella here. So the innermost layer is the secondary wall. Okay, so that is it's strange because you think secondary is maybe something that's further on, you'd have primary and then secondary, but it's the other way around. Yeah, so it's good to know if you're studying the biology of plant cells. Okay, all right, so after that, if anyone has any questions about plant cells and their mitosis, let me know. If not, I'll move on to the next technique, which is a very well established high throughput screening technique that was derived in yeast cells, and it is used to detect protein-protein interactions. So how does this work, right? So we have um, this reporter gene. Okay, this is the blue one, that's the reporter gene, and expression of it depends on a transcription factor that has two domains. Okay, one is and these two domains, they both are needed to bind to the promoter elements to activate reporter gene expression, but these two are actually quite functionally and structurally independent. They are bound to one another normally in normal yeast or in a normal condition where you're not doing any assay. Typically this DB and AD, they are bound to each other. But what scientists did is they were like, oh, because these two are so functionally and structurally independent, we can actually separate them out and use them for our experiment. So the DB or the DBD, that is the DNA binding domain. That's the one that actually binds to this region in the DNA, in this UAS portion. And then you have the activation domain. That is part that interacts, that is required here as well for activating the transcription of this gene. So you need both of these components to be able to transcribe this gene successfully. So what did scientists do? They took the DB domain that binds to this DNA sequence and the AD, which activates the gene expression, and they separated them out. They're normally fused, they are bound by some region, but because they're independent, they separated them out. Then the DB domain you can be fused with a protein that you can say is called the bait, while the one fused to the AD domain, that is fused to the prey protein. So you have a protein bait and you have one as prey, and that's one is fused to DB and the other is fused to AD. And now we want to know whether prey and bait interact with one another or not. So what will that tell us? How will we figure out if they're interacting? If they interact, then DB and AD will come into close proximity, and that will allow reporter gene expression to take place, right? And if they don't interact, they won't be there near one another, and the gene will not be expressed. Simple enough, right? So that must be clear also, and that was explained nicely in the coursework. So there are actually some uh, sort of alternative or adapted ways of uh, looking at other kinds of interactions also using similar yeast to hybrid system setups. So I thought I'd go into those as well because they're useful to know about. So the first one is the yeast 1 hybrid system. So in the yeast 1 hybrid system, in this assay, it looks actually at your protein DNA interactions. So what is done here is that you have this reporter gene and some target sequence. And we want to know whether, so you would need to make sure that you clone this target sequence upstream of the reporter gene. So not too far away. And we want to know whether this prey protein is binding to the target sequence or not. So this target sequence is DNA and the prey is some protein that we want to know if it's binding with this. So what do? We fuse prey protein with AD, the activating domain. This target sequence is over here. And if this prey protein is interacting, then this AD will come close to the reporter gene and that will be enough to activate the reporter gene expression. So this is the yeast 1 hybrid system. Okay. Next is a yeast 3 hybrid system. This is another sort of adapted method here, which looks at protein interactions mediated by a third component. So this could be an RNA molecule or some other protein. That depends on whatever you're looking at, whatever experiment you have planned. So how does this happen? There's a further intermediate here. So here bait and prey, they don't actually interact with each other directly. What they do is they bind to some other molecule, some intermediate molecule in between. In this case, it could be an RNA molecule, for instance, that would have some different sequence specificity that kind of depends on what it's interacting with. So what would happen is bait is bound to your DNA binding domain, prey is bound to the activating domain. And if this, say, RNA molecule were to come and bind and bring these two together, then only we would have DB and AD coming close to another and again reporter gene expression. In this case, the reporter gene can be the beta-galactosidase enzyme that can have a coloring effect with X-gal substrate, right? So I hope that is clear for everyone. These different types of yeast 1 hybrid, yeast 2 hybrid, and yeast 3 hybrid systems. There's one more type actually. This is called the split ubiquitin yeast 2 hybrid system. So this is a yeast 2 hybrid that was adapted for insoluble membrane proteins. So if there are two proteins that are in the membrane, so they're not just floating around freely in the cytoplasm. So typically if there are two proteins that are deeply embedded in the membrane, they have a lot of hydrophobic regions that allows them to be amongst all of these lipid tails, right? So we won't be able to assay them in the cytoplasm particularly easily. So how do we do this, right? So what we do is, suppose we have a bait protein that's in the transmembrane and we have a prey protein that's in the transmembrane. We want to know if these two are interacting. We can fuse bait with this cub domain. So cub is a C-terminal ubiquitin moiety and prey we can fuse with nub, which is an N-terminal ubiquitin moiety. Now these two, if they fuse with this transcription factor, if they interact with one another, they actually get cleaved. And cub has this transcription factor AD and DB. This is the full transcription factor that's required for reporter gene expression. So this entire transcription factor is attached to cub. But this region that's linking cub to the transcription factor has some sites where a protease can come and cleave this. And it will only do so when cub and nub are interacting. So the interaction of these two query proteins, bait and prey, will allow cub and nub to interact with one another, which is then recognized by this protease that comes and snaps it. Then DB and AD, the complete transcription factor, will go and translocate into the nucleus, bind to the regulatory element and allow the transcription of this reporter gene. So by that way, you will be able to tell whether two transmembrane or non-soluble membrane proteins are interacting with one another. Okay, so I hope that is clear. Um, so does anyone have any questions about this technique or any of the yeast hybrid techniques? Cool. So this is the original paper on yeast to hybrid. It's really nice to read because it's such a short paper and it's so informative even though it's so short. And it nicely demonstrates this technique. This is what I was showing you earlier in those diagrams, right? That you have these two parts coming together, binding to UAS and allowing the LAC-Z gene to be transcribed. So this is the technique that they have sort of outlined in this paper that came out such a long time ago. So if you're interested, do check it out. It's a nice read. Okay, so I have a few more questions for you guys. So the first one is, what is the primary purpose of the yeast to hybrid system? What kind of interactions do we study using yeast to hybrid system? Yeah, I have one answer. Does anyone else want to try? Yeah. Yeah, looks good. I think everyone's getting it. So it is B. It's to study protein-protein interactions. And just to revise, your yeast 1 hybrid looks at protein DNA interactions, and yeast 3 hybrid looks at maybe two protein interactions mediated by a third element. It could be RNA, it could be protein. And your split ubiquitin yeast 2 hybrid looks at protein-protein interactions, but those proteins will be embedded in the transmembrane domain and typically quite hydrophobic, okay, or insoluble. All right. In the yeast 2 hybrid system, transcription activation occurs when? Which of these following? Okay, I have one answer. Anyone else wants to try? Yeah, so the answer here is A, where you have bait DBD, bait is bound to DBD, and prey AD, these interact. So maybe it's a little bit confusing because I've written bait, prey in B, but here that means DBD and AD are bound to one another. But that's not the case, right? They are not bound to each other. So bait and DBD and prey and AD will interact via bait and prey. That will bring the activation domain closer to the promoter and transcription activation will occur. Okay, so then you will get these blue colonies. So I hope that's clear for everyone. Okay, next question. Which of the following is an essential negative control in a Y2H experiment? Can you guys tell? Anyone? So in an experiment, say you're doing some experiment, you're doing a PCR, okay? You're going to have some controls always to make sure that you're doing your experiment correctly and whether the results that you're getting are not something arbitrary that might have happened by mistake, right? So a positive control is something that will always work. So suppose you're doing a PCR and you're checking whether a gene is there or not. Okay, so you have some samples and you want to check the gene is there or not. So a positive control would be a sample where you know the gene is there. That way you will always get a band or you will always get a positive signal from that sample. So that will tell you that, okay, my assay conditions are fine. Everything that I've added, there's no contamination because I'm getting the band. But what is one more way of knowing that your assay is fine? That is the negative control. That is to make sure that this positivity is not coming from some other contamination. It's actually a true positive. That would be a negative control where you say you don't add sample only. You have everything else needed for your PCR. You just don't have your sample. In that case, if you get some random band, you know that there is a contamination. But if that lane is completely clear, you're not getting any signal from that sample, then you know that your negative control has worked and you validated your assay. Right? So in this kind of an experiment also, you would need a negative control also. Maybe if you're setting up a new Y2H experiment, a new yeast-2 hybrid experiment, you would need to make sure that there is no self-activation. Because what might happen is that there might be some low level baseline transcription activation that's just happening, right? What if what if DBD and AD are just coming close to one another at baseline level and causing activation? So you need to make sure that there is no self-activation at the baseline. So that's why the answer here is A. If you bind BAIT to DBD alone and you test it out, you can check whether on its own is it binding with AD and causing self-active or causing transcription activation without any prey involved, right? Because cells will have some amount of AD in it anyway, right? So BAIT and DBD alone to check for self-activation will give you the baseline of what is above which you will see an interaction or above which you will see a transcription activation. So that's an essential negative control, that you should not have too much self-activation, so that you know that when bait and prey are actually interacting, whatever signal you get should be much higher than whatever is the baseline. So I hope that is clear. And B is not right because AD will obviously bind with the DNA. In the sense, it will bind with the DNA with the help of DBD, but it doesn't have to. AD doesn't bind to DNA on its own, right? So checking if it binds to DNA is not going to help because it doesn't bind to DNA. So there is no reason for pre-AD to bind to DNA. So that's not a negative control. If it were to bind and it gives a transcription, then it might be a positive control. So yeah, I hope that's clear. Okay, so with that, I will move to the next topic, which is about induced pluripotent stem cells. So as you all know, this uh, amazing um, discovery that you can induce pluripotency into adult cells, so essentially make stem cells from adult cells, was by Shinya Yamanaka and his postdoc Takahashi, and many others who had found out about these different factors also before him. And so then, he did some really great experiments and found out about these 4 factors that are involved in inducing pluripotency. And he got the Nobel Prize in 2012. So, as you know, these embryonic stem cells, which are derived from the inner cell mass of blastocysts, they have the ability to grow indefinitely. So, grow continuously for a very, very long time while maintaining pluripotency. So, pluripotency means that they can essentially divide and differentiate into any kind of tissue. They can become heart cells, muscle cells, bone cells, neural cells, in anything. Okay, so that's pluripotency. And the ability to differentiate into cells of all 3 germ layers. But yeah, and human embryonic stem cells, of course, that means that they could be used to treat any sort of diseases. These degenerative diseases where you start losing a functional pool of cells and you want to replenish them. So like in Parkinson's disease, Alzheimer's disease, spinal cord injuries, and even diabetes, where you have lost a good number of your islet cells. you'd want to replenish them maybe with these embryonic stem cells. But the issue here is that there are so many ethical difficulties naturally, right? Because how can you use human embryos for so much, so many different kinds of experiments and reviving all these different people? So the problem of tissue rejection also, of course, is there, that not everyone has a human embryo with MHC compatible to their body. So how will you do this transplantation of, say, some human embryo cells into some completely unrelated patient? They will have immune rejection. So cells from your own body that are pluripotent, that are then put back into your body to then regenerate your cells. That would be the best condition, right? But how do we do that? We don't have pluripotent stem cells just hanging around in our bodies that we can culture and then inject into some wound or something, right? So one way to circumvent this is, of course, generate pluripotent cells from the patient's own cells, so somatic cells, adult cells. And so somatic cells can actually, and this was an experiment that was shown to you in the coursework, that somatic cells can actually be reprogrammed by transferring their nuclear contents into oocytes or by fusing it with embryonic stem cells, indicating that these unfertilized eggs and embryonic stem cells have some factors in the cell, in their cytoplasm, right, that can confer totipotency or pluripotency to somatic cells. So if you have the nuclear contents of a somatic cell, those genes related to the somatic cell will be transcribed, and this cell will become pluripotent or totipotent if it has the factors that typically are expressed in high levels in these embryonic stem cells or in oocytes. Okay. So this was known at the time. But again, it's difficult to get an oocyte or these embryonic stem cells. So they had to figure out what is it that's there in their cytoplasm that can transform a somatic cell like that. Okay. So they hypothesized that these factors are there and they play an important role in maintaining ES cell identity and also play important roles in inducing pluripotency in somatic cells. And so with some very clever experiments, they demonstrated that they could induce pluripotent stem cells from mouse embryonic or adult fibroblasts, so somatic cells, by introducing these four factors, OCT34, SOX2, CMYC, and KLF4 under ESL culture conditions. So I thought I'd go into a little bit of this paper because it's such a, really great way to learn. And it's very interesting to see how, in the simple way in which they did experiments, that then brought about such an amazing discovery. Okay, so this is the paper. It came out in Cell in 2006, and this is what it is. They're inducing pluripotent stem cells from mouse embryonic and adult fibroblast cultures by defined factors. Those four factors, which are called Yamanaka factors. Okay, so what did they first do? Okay, they selected 24 genes as candidates for the factors that induce pluripotency in somatic cells. So remember, they're trying to figure out what are these different factors that are present in these embryonic stem cells or um, embryonic stem cells or oocytes that essentially make them able to induce pluripotency. So there was already information about some genes that might be upregulated or some candidates that might be there. So they decided that let's comprehensively go through all of these candidate genes and basically do like permutation combination kind of thing to see which combination of genes is required really for inducing pluripotency. So they developed an assay system. I'll just explain it in brief. And this is basically what an assay system that could check if induction of pluripotency has happened or not. And if it has happened, the cell that has been induced to become pluripotent would suddenly have resistance to G418. G418 is neomycin. It's basically an antibiotic. It's like this is an antibiotic, neomycin. It can um, kill mammalian cells, okay, this antibiotic. But if you have the resistance to G418, then the cell will thrive. It will not die. It will be able to nicely live on. Okay. So what they did is they inserted this beta-GO cassette. This was basically a fusion of beta-galactosidase and neomycin resistance genes. So not only will they have beta-galactosidase resistance, sorry, beta-galactosidase expression, which can then, if you have a positive colony, then this entire construct will be expressing, meaning that you will have beta-galactosidase that can act on X-gal and give you blue colonies. Also, you will have neomycin resistance, meaning that it will survive even if neomycin was added into the culture. This cassette was in the mouse FBX15 gene. Now, this mouse FBX15 gene is expressed specifically in mouse embryonic stem cells, meaning that if the cell has been uh, correctly induced into pluripotent state, this gene will start expressing, and these two things, both this beta gal as well as neomycin resistance, will also start being transcribed, meaning that the cell, if it has been induced into pluripotent state, will be able to produce beta gal and also be resistant to neomycin. So that is the way they were screening and telling, okay, this has been induced into pluripotency at some level. So normally your embryonic stem cells that are homozygous for this kind of construct were resistant to very high concentrations of G418, up to 12 mg per mil. So typically in culture, when I select cells with G418, not stem cells but other cells that I have transduced with something, I don't go above like 2. So 12 is like really high, okay? So high concentrations of G418 up to 12 milligram per ml, whereas somatic cells derived from this beta-geo mouse were sensitive to normal concentration. So what they predicted was that even if they have some partial activation of this FBX15 locus, again, this is specific to mouse embryonic stem cells, meaning that if induction of pluripotency has happened, this gene is going to be at least partially activated. There, it would result in some amount of resistance to normal concentrations of G418, like this. Okay, so that was their readout. That's what they wanted to see, whether there is resistance or not, and also to visualize the colonies. They did the beta gal. And this is what it looks like. You have your FBX15 locus. If it's not induced, then it will not express. If it's induced, then it will express. And how are they inducing this? So they're doing a retroviral infection. So basically, long story short, they have a virus in which they have the gene of each one. So there are 24 different retroviruses. Each one will have one of these candidate genes. Okay, and they are doing in 24 different cases, 24 different wells of cells, they're adding this retrovirus to each one and seeing, with the help of G418 selection, is that factor enough to induce pluripotency or not. Okay, so here what they did, so they did individually 24 candidate genes, one, like that, not mixing together. They didn't see any pluripotency. Okay, that I haven't shown that figure. Here they have added all 24 candidates together. So they use the retrovirus for all 24 of the candidate genes together in these cells. And what they saw is that they got 22, if they counted, they saw 22 G418 resistant colonies, meaning that there are 22 colonies where some or all of these viruses have gone in and induced some amount of pluripotency. And this is what they saw. They did further colony screening after that. They looked at the colonies actually and saw, okay, how do they look? Do they look like embryonic stem cells? Do they look different? And you can compare. These are mouse embryonic fibroblasts. These are normal fibroblasts. These are embryonic stem cells. And these are mouse embryonic fibroblasts that were infected with a combination of these 24 candidates. And in this case, you can see how their morphology has changed, how they've become sort of more rounded, and they're forming these clusters. And so they determine this to be much like the embryonic stem cells, meaning that these have been induced with some level of pluripotency, definitely. Now, one issue is that when you're adding all 24 candidates together, we don't know whether all 24 viruses got into the same cells and whether all of them are necessary or not. So we need to further go in and see which of these 24 candidates are actually needed, right? So a combination of G418 resistance as well as your screening based on the morphology of the cells and the doubling time of the cells, which is, they're basically comparing to normal embryonic stem cells. They use that to try and then narrow down further on which of these 24 candidates it is. So how did they do that? So the first thing they did is to narrow down out of these 24 candidates. So they examined the effect of withdrawal of individual factors on the formation of these resistant colonies. So initially, what did they do? They have all 24 of them. From each one, they are minusing 1. So this is 23 of the factors minus factor number 1, 23 of the factors minus factor number 2, right? 23 of the factors without this factor number 3. So out of the 24, at each time they're taking out this factor number 10, 11, 12, like that. Okay. And of course, you have the control of all of the factors and the mock, where you don't have any colonies forming at all. Mock is your negative control, okay, where you don't add any viruses. So you're not getting any kind of pluripotency. So what did they see after doing this very extensive experiment? They saw that initially there were these 10 factors, the numbers are listed here, whose individual withdrawal from the bulk transduction pool resulted in no colony formation and at day 10, and very few colonies formed at day 16. So these ones basically are highly essential, right? So we are figuring out that, okay, out of 24, eliminate 14, these 10 are essential. They have to be there. Without them, the others cannot do the job. So next, what did they do is within these 10 factors, they narrowed in further. So they looked at morphological characteristics of these resistant colonies, and eventually after a bunch of other experiments, where here they also did this thing where they did the 10 factors minus 1, minus 1, minus 1. You understand how it's going, right? And then all 10 factors as the control, all factors as another control, and mock. And then they again looked at colony factors. So when they looked at this initially, they found out that the morphological characteristics of these resistant colonies coming up here indicate that OCT34, KLF4, SOX2 and CMYC seem to be playing very important roles in generating iPS cells. And then they comboed these 4 genes and they tried minusing each one. So in this one, they didn't put CMYC, they put the other 3. They didn't put KLF, they put the other 3, SOX, and so on. And so you're seeing that the best um, case where you have the most number of colonies and also the most number of sort of morphologically accurate colonies actually comes up in this one where they have all 4 of the factors. So all 4 of these are highly essential. Okay. And this is very similar to 10 factors, meaning that the other 6 in this case were probably not that useful. It's these 4 that are uh, the main ones. Okay. So with that, they figured out that it's these 4. So that's very good. Now they've generated iPS cells. That's wonderful. But are these iPS cells actually able to form any tissue that they like? So how did they do that? They examined that the pluripotency of these iPS cells by using teratoma formation. Teratoma is basically just a big mass of cells that grows and grows and grows when you have injected something. It's like a solid mass of cells. And they observed that in these teratomas, after injecting iPS cells into a mouse, they observed all 3 germ layers were forming from most of the clones. So you can have different clones, right? If you have like 7 different colonies, each colony is a clone. So you want to test and see which one is the best clone. All of them, of course, are expressing all 4 of these factors. You're just testing between clones to see which one is the most efficient. So most of the clones that they injected seem to have all 3 germ layers. So you have cartilage forming, they did some histological staining. You see that this is their central nervous system. Muscles are forming, adipose tissue is forming, right? Fat tissue, epithelia are forming. So you have all of these different tissues that are forming from the same from these adult fibroblasts that had been induced to become pluripotent. So that is the huge thing that they've done. They had many other experiments also. They did whole RNA-seq and so on and so forth. Lot of things to confirm that what they're seeing is not something crazy, but actually a true result. But I thought this would be enough to sort of show you guys um, how this work was done. And it sounds so simple, right? You just don't use one and see, then take one out and then see again. But it's a lot, and it's a very interesting paper. So I'd encourage you guys to check it out. The link is over here. Yeah. So if anyone has any questions about this paper, please feel free to ask me, or you can look up this paper in your own time and read it and see. It's a pretty easy read, and it's quite interesting. Yeah. Cool. So that was all about your iPSCs, and I have questions about stem cells and stuff later on. But maybe we can jump to the next topic right now if no one has any questions. I hope you guys are all still there. Yeah. So in your, I think, third lecture, you guys learned about how these fluorescently labeled iPSC lines were used to study some major structures in the cell, right, by a guest lecturing professor. So the essential sort of pipeline that they followed in in their work was that they initially have some established human iPSC line, and then they gene edited it, right? So they introduced a fluorescent protein at the locus of the gene of interest. So what does that mean? The gene of interest, suppose I want to study the mitochondria, okay, and I want to look at some mitochondrial membrane. So some mitochondrial membrane protein, I want to label it with a fluorescent protein so that I can look at the mitochondria under the microscope and see it fluorescently labeled, right? So this cell, wherever the gene of this protein in the mitochondrial membrane is, there I want to introduce a fluorescent protein that will essentially express alongside the mitochondrial protein. So I need to determine the locus, that means where the gene of interest is within the genome of this iPSC, and then do my gene editing there and introduce the sequence that codes for the fluorescent protein into that locus or at near or at that locus. Okay, that is how I will label mitochondria, or say anything, microtubules, actin, whatever you like. Okay, that's how I will label it, by editing directly in the genome in a way that I know where I'm adding the gene. And this is they did using CRISPR-Cas9 technology. And the way they did it, I think they used HDR based insertion, and I have a slide on how that is done typically I think coming up soon. And once they have edited this cell line, the gene of the cell, uh, then they are going to essentially make sure that they have a pure culture of them. So they're creating a clonal line generation. So what do? Is they check that the cell line has integrated this gene homogeneously, and they'll sort out the cells only that are expressing this fluorescent protein. So they don't want any cells that are not expressing it. Once they do that, they've made a stable cell line expressing this fluorescent protein, and then they can use that to do different experiments. They can do live cell imaging, where you look at the cell in a live chamber, which typically you can warm and humidify, and you can image the cells while they're alive, maybe interacting with something or reacting to some treatment that you have given. And so they imaged both in undifferentiated cells and differentiated cells. They differentiated them to get different kinds of lines. So one was that you saw the beating hearts, right? The beating heart cells. So those were the cardiomyocytes that they differentiated these cells into to get that. And that's a great model to study, right? Getting cardiomyocyte stable cells from a human being is going to be very tough to do, and it will not last for very long. But a cell line that is stably expressing a fluorescent protein, if you can differentiate it and say I want to study microtubules in cardiomyocytes, I can do that, right? I'll have an iPSC cell line, human iPSC cell line that has the gene edited form of it, shows a fluorescently labeled microtubules, and I will differentiate it into cardiomyocytes. So now I have cardiomyocytes that are going to last for quite some time, and they have fluorescently labeled microtubules. So that makes things a lot easier, right, than going to a human and taking out their cardiomyocytes. Um, and of course, they look scale up for the distribution of the cell line. So if they want to, sell it or distribute it to other scientists who want to do this kind of study. So this is some really interesting work that they've done and doing currently. And I thought I'd go into a few of the techniques involved here, which would maybe help you understand how they perform these different steps in their pipeline. So the first thing, I don't know, have you guys studied too much about CRISPR-Cas9 technology? I'm not going to go into it in like very great detail, but what it involves typically is um, this Cas9 protein and a guide RNA. So what is this? Where did this CRISPR-Cas9 technique come from? So it was seen that bacteria also seem to have an adaptive immunity. So you guys know, from immunology that um, people, us humans, even plants actually have an adaptive immunity where we're able to detect uh, some uh, some pathogen, maybe a virus or a bacteria, and we are able to we're able to detect it specifically in an antigen specific way, and then T cells or B cells can respond to it and can have an immune response that can neutralize this target and protect us from disease. So bacteria also have such an immunity where they're able to detect and cleave up viral proteins or bacteriophages that enter the bacteria and try to take over. So they're able to protect themselves from bacteriophages that are trying to kill them, right? So looking at that, scientists realized that that might actually be a great way, the technology that already exists in bacteria, if we just adapt it a little, it might be a great way for us to be able to insert genes or cut genes where we want to for gene editing purposes. Okay. So in your CRISPR system, you will typically have two main components. One is your gRNA or your guide RNA. Okay. So suppose this is my gene of interest, this green color. My guide RNA will have a region that is complementary to it. Okay. So that is what is going to specify. The guide RNA will go and bind there and specify that this is where the protein should, Cas9 protein should come. Now Cas9 protein is going to create a double-stranded break. So this guide RNA will bind here. There is a region in the guide RNA that allows Cas9 binding. Okay, so it goes binds here, calls Cas9. Cas9 comes and binds here, and so it's a short synthetic RNA composed of the scaffold sequence. It's necessary for Cas9 binding, and also this region, these nucleotides. This is what will define which target in the genome is going to be modified. So this is the target or the scaffolding spacer region, and this part is what binds to Cas9. Okay, and this PAM, this is called a protospacer adjacent motif. This PAM site needs to be very near where the Cas9 is going to cut. And this sequence will depend on which Cas protein you're using. So I think in Cas9, it's like NGG or something like that. I'm not completely sure. And the gRNA will have a sequence that is unique compared to the rest of the genome. Why is it important that this sequence is unique, that it doesn't bind to other stuff? Because then it can go and cleave anything, anywhere, right? It can shear off the whole genome if it's non-specifically binding. So you want to make sure that this does not have hits anywhere else in the genome. It is majorly binding here to the gene of interest where you actually want to do your editing. And second is that the target is present immediately adjacent to this PAM sequence, which is very important for Cas9 binding and to the right target and cleaving. Okay. So these things you have to keep in mind when you are designing guides. So you can design guides using, there is this software, or not software, it's like a website created by Sanjana Lab called Guides, GUIDES, and that's something I've used before to make guides for this. And it essentially tells you how much off-target binding is there and how good of a guide have you created, how much on-target binding is there, how well is it binding to just one spot and nowhere else, right? Its specificity, it will be able to tell you, as well as other characteristics. So that is the main, those are some of the main components of CRISPR-Cas9 system that you would need to do a knockout or a knock-in. Okay. So how is a knockout done? You will have your gRNA. This spacer is what binds to your target in the genome. Scaffold is what binds to Cas9. Now you form this complex, and this Cas9 sgRNA complex will then go and bind to the target. The target is there with the PAM site. Cas9 will bind there and it will cause target cleavage. So it will cleave and create a double-stranded break here. Now we know, from our previous studies that when there is this double-stranded break, there are some repair pathways that come into play, right, to fix this. So one of these is non-homologous end joining. So this is a quick pathway and it's quite error prone. So what will happen is in the wild type case, it will join it and that's all. No harm, no foul. There's no mutation there. This gene is fine. Other cases is there will be some inaccuracies while this non-homologous end joining is taking place because there is no template here, right? It's just joining. So there can be an insertion here, an insertion mutation. There can be a deletion mutation where we have got rid of some of the gene by mistake. There can be an entire frame shift mutation, meaning that this entire region is gone, meaning that gene is now not the correct number of codons. So what will happen if there is a mutation in this, some kind of mutation in the gene after CRISPR-Cas9 based editing is that the gene might become non-functional, right? It will not be, either it will not be transcribed at all, or if it is transcribed, it will create some nonsense product that might be either sent off for degradation or it will just not have any effect in the cell. Because, typically you will make sure that you're cutting up a region that is functional to that protein. So suppose you want to, in a cell, there is some protein that's there in the nuclear, inside the, whatever, in the nuclear pore complex, you want to get rid of one of the proteins. So how do you do that? At the locus, somewhere within that gene, some useful functional unit of that gene in some exon typically, you will go and you will design a guide against that and you will make sure that the CRISPR-Cas9 and guide RNA complex will then go there, cut that functional domain and make that entire protein non-functional. So either it's not transcribed at all and then therefore not translated into a protein at all, or whatever is produced is some junk and it is not useful to the cell. So in that way, actually, you can completely knock out that particular protein from your cell. So this is genome editing, meaning it's not a plasmid that is episomally, sitting around in the cytoplasm that you're doing. So if you have edited the genome and you grow that clone which has got that knockout, then all the cells are also going to be knockouts, right? You have removed that gene entirely from the genome, so any other daughter cell that comes from that cell is also going to be a knockout. So making sure that you have a pure clone and a pure colony from, a clone that has proper knockout is very important in these cases. Okay. So I hope this is this is sort of comprehensible to you guys. It's digestible. So then that was knocking out, meaning you're removing a gene or you're making something non-functional so that you can study it. Another way of using CRISPR-Cas9 is actually knock-in. So knock-in means that we are putting a gene into the genome. This is something that this professor had shown, right? That they are knocking in this fluorescent protein, meaning that now they are going to make it constitutively expressed from the genome from a particular locus of a gene of interest, right? So how do you do that using CRISPR-Cas9? So this is a very clever way. You can create this template. This is a little nifty to create, but once you've done it, it's pretty great. So this specific change, what they have called here, this is your gene of interest. So say this is your fluorescent protein. Okay. So on the left, you will create a left homology arm. Right, you will create a right homology arm. So what are these homology arms? They're basically regions that are complementary or that will bind to a locus in the genome where you want to insert this gene. So suppose this gene is for microtubules. Okay, this entire gene is some functional region of the gene of microtubules. So I want to label microtubules with GFP. So this is GFP. This left homology arm corresponds to this region. This right homology arm corresponds to this region. Typically, it's like 400 to 500 base pairs long. And this is the fluorescent protein. Now what I will do is I will have some site here, some guide RNA that will help bind to this region over here that I have specified. And Cas9 will be called here and it will induce this double-stranded break. And this is bound here and this is bound here. So I'll have my gene of interest of fluorescent protein in a loop over here. Now what will happen? Earlier you saw that there was NHEJ happening, right, which is inaccurate and can cause a lot of these different kinds of mutations in the case of knockout. Here it actually encourages HDR, which is homology directed repair. What does that mean? Essentially, the cell has found a template with which it can help to repair this region. This template in this case is my fluorescent protein. So what happens? I have made the cut here, and I have had a complementary ligation of the left arm and the right arm to this region. So I have my template of my fluorescent protein kind of chilling over here. And what happens is, with the help of different repair enzymes, actually it uses this homology, this template of the fluorescent protein to insert bases into the genome here, and that actually creates this fluorescent proteins gene in the genome. So by cutting this and providing a template of the gene of interest, we're actually integrating the gene of interest into my uh, double-stranded break region. And so this is how it looks finally. This is the mRNA that is created from the gene which had introduction of the particular change or fluorescent protein. So by this technique of HDR repair, using a cut initially created with Cas9 and a gRNA complex, with this technique, we're able to then knock in fluorescent proteins to mark whatever we like. So I hope that's clear. So that's how this professor and others in her team were able to knock in those fluorescent protein at specific loci and label proteins of interest so that they can study them in different techniques. So is that clear to everyone? CRISPR-Cas9, the basic technology, and how knockout and knock-in is done using a Cas9 gRNA complex, and how HDR uses a template, which is actually just the gene of interest that you want to insert into the genome. Is that clear? Okay. So great. So, all right. So now one might ask the question that she has shown, right, that here you have gene editing where you have this CRISPR-Cas9 going in, right? So, or you saw in the Yamanaka paper that they had made retroviruses that had each one had one of those factors, right? The gene of one of those factors, and those retroviruses were being used to infect the fibroblasts and sort of integrate that gene into their genome and allow its expression, which can then allow us to study it. So we're overexpressing some gene, but how are we getting the gene into the cells in the first place? So there has to be some methods, right? So I thought I'd go into some gene delivery techniques that are used um, oh, to introduce foreign genes or genes that we want to overexpress to study some particular um, function in the cell. So I thought I'll go over this. So one thing is that when you're performing gene delivery, if you're performing this via a virus, say retrovirus, adenovirus, lentivirus, adeno-associated virus, whatever, that is called a transduction. Okay, so in the lab, if someone says I've transduced my HEK cells, that means they've added a virus to their HEK cells that has then integrated with the HEK cells genome and is stably expressing that gene of interest that was there in the virus. The other mode of delivery, gene delivery, is non-viral DNA, and that is called transfection. So that is typically using plasmids, plasmid DNA, or you can even have some oligos and stuff, and you can sort of transport them into your cells using a non-virus method. So viruses, as you know, have ways of entering cells on their own, right? Because they're made to infect cells. So they bind to certain receptors on the cell and are able to inject themselves in. Transfection is where we use other techniques to get the DNA into the cell so that it can express. Typically, it's harder to have it completely integrate with the DNA and make a stable, but you can still do it. So in viral delivery, you have these biologic ways where you have retroviruses, adenoviruses, adeno-associated viruses, lentiviruses. Okay, these are different types of viruses with certain edits and all. Like whether they like retroviruses and lentiviruses differ in that fact that retroviruses can infect only uh, dividing cells, whereas lentiviruses can infect both dividing and non-dividing cells, things like that. So based on some elements that you add into the viral plasmids. And for non-viral techniques, of transfection, you can have chemical and physical methods of introducing DNA or gene delivery, right? So chemicals can include polymers, or it can like PEI, or it can have liposomes. You guys might have heard about lipofectamine, right? Uh, and then physical techniques, there's gene gun or biolistics, microinjections, electroporation, I think even sonoporation. These are different techniques in which you can make transient pores or inject the DNA directly into the cell, um, without using a virus. So some chemical transfection methods are these cationic liposomes that I mentioned. These liposomes, they're cationic, meaning they have a positive charge. So they can bind to these DNA which have negative charge, and they make an uh, overall positive charge on this complex, which helps it bind to the membrane, which is typically negatively charged. And that helps it then get endocytosed into the cell. Another method is calcium phosphate. So this is using calcium ions, which are, as you know, divalent and positive. And they can also bind to the DNA and help with attachment and cellular entry, where it will get endocytosed into the cell. If something is stuck, the cell will make an invagination here and it will endocytose whatever is stuck on its surface into the cell and process it further. And you can also have complex formations with different chemical reagents. One common one is this polymer called PEI. I use that a lot. It's another positively charged or whatever cationic polymer that can bind to your DNA, and based on your requirements, you can use branched PEI, you can use linear PEI, whatever you require. That can make this complex bind to the cell surface, and then it will get endocytosed and allow for eventually, when the the DNA escapes from the endosome, it will enter into the nucleus, and then it will be transcribed, and it will then the mRNA will be formed, which will be transcribed and then translated, and the protein will be expressed or overexpressed in the cell. So what does that mean? That suppose this cell has some normal amount of actin, but we want a fluorescently labeled actin. So we can overexpress a fluorescently labeled actin probe. So we use this actin probe in a plasmid. We can either use a liposome or this calcium phosphate method or cationic polymers to complex that plasmid DNA, which will come and stick onto this. It will endocytose, and then it will produce high amounts of that plasmid DNA. And that fluorescently labeled actin will be produced, and then we'll be able to see cytoskeletal dynamics using fluorescently labeled actin. Right? So these were some chemical transfection methods that is used to introduce foreign plasmid DNA into cells. There are some physical transfection methods also. One example is electroporation here. This is essentially you use an electric field across the cell, and that creates some transient pores. So here you have a cell membrane that's intact, and we want to insert some DNA CRISPR-Cas9 um, protein and the protein DNA complex into the uh, cell from outside. So what do? We have the cells in solution along with this DNA or protein or whatever. We apply an electric field across the membrane, or we can give it a pulse, or we can apply it for a longer time depending on what kind of cells you have or what kind of experiment you want to do. And these transient pores form, and this DNA or proteins or RNAs or whatever, they will enter into the cell. They will enter directly, so they're not endocytosed in this case, and then eventually they will be expressed here. But one issue with this technique actually is that if you're not careful, it requires some amount of standardizing. If this electric field is too much, it can actually completely fry the cell, and if it's not enough, then nothing will happen. So you have to play around um, with these parameters a little bit to get a good amount of DNA to enter into the cytoplasm and nucleus. Yeah. So that was the physical transfection methods. And finally, of course, there are the biological transfection methods or viral vectors, which is the actual technique is called transduction with your viruses. So typically what do? You start with some plasmids. These plasmids can be of your, they are your plasmids of interest, which will contain your gene of interest as well as some packaging components. So in this case, they've shown a packaging cell, but that means a cell that already has the packaging components integrated in its genome. You can also have packaging plasmids. So what does packaging mean? You have these different uh, components of a virus, right? That make up its capsid, that makes up the receptor on its surface and stuff. So these components are also encoded into separate plasmids. And if you put the gene of interest along with those, then it essentially creates a virus with all of those components, so a complete virus, as well as your gene of interest as a part of its genome. Okay. So a virus expressing your gene of interest, a complete virus, but that is only possible from all of these plasmids. So one might ask, why can't I just have everything on one plasmid? It's a matter of A, to size of plasmid, and B is safety. Okay. And so you transfect these plasmids into a cell and produce virus. Okay. When you produce virus, you concentrate that virus, you check its MOI or you titer the virus to see how much of virus to add to how much of cells. So the potency of the virus that you have produced, that you can check. And then so now you can see here, right, that these different plasmids are producing different components. So it's assembling within the host cell when you have transfected it with these different viral plasmids, and they make one complete virus over here. And this virus is then used to, after concentrating and so on and so forth, you use it to transduce your cells of whatever it is. Say you package these typically in HEK293T cells. From this you take the virus, and that you can use on whatever cells you want. So maybe I'm using this on some, I don't know, some lung cancer cell or something. Alright. So I use this virus on this lung cancer cell and I express my recombinant protein that's within this viral genome, and I overexpress the protein. So that's how it's done. So in this, I think they're showing a lentivirus, which is a very commonly used mode of transfecting cells, particularly adherent cells. Okay, and this DNA will then, from the virus, can integrate into the host genome and produce the protein. Because as you know, a virus needs the host to be able to produce proteins that are encoded for in its gene, right? Genome. So that's why it will integrate, and you can actually make it stable, meaning it will continue to express and it will integrate as part of the genome of that cell itself. So you don't need to do anything else for it. So I hope that was clear. Another technique I thought I would go over is flow cytometry and FACS. So as you saw earlier here, they produce these clonal lines, right? So now suppose they have a fluorescent protein, you can see this. Now some cells might not have it also, right? So you might have a population where, say, just by looking at it under the microscope, some 40% of your cells are expressing the fluorescent protein and maybe 60% or so are not expressing it. So I want to separate out only those which have fluorescent protein and only those that and separate them from the non-expressing one. I want to have a pure culture of fluorescent protein expressing cells. How will I do this? So firstly, how will I check how many cells are positive? And second, how will I separate them out? So one technique that she mentioned they use, and this is a very, very, very common laboratory technique that people have to do in their studies, is flow cytometry and followed by FACS. So I thought I'd tell you how that works as well. Yeah. So have you guys heard about flow cytometry and FACS sorting before? Anyone? Just let me know. Or have you done maybe flow cytometry and FACS sorting? Anyone? So I'll explain how it works. I just want to know how much detail I need to go into. I guess maybe you guys have heard about it in your studies and all, right? But okay. So I'll just explain it from a practical point of view then. So flow cytometry is a very important technique that can tell you a lot about your cell sample. It can assess properties like the size of your cells, the granularity of each cell, and also, of course, the fluorescent signal from the cell. But in flow cytometry, you're not actually saving the cell sample. So suppose I have a culture of some fluorescent protein expressing cells and some that are not expressing. What I can do is I can take these cells, suspend them in some buffer, okay, some flow cytometry buffer, and pass them through the flow cytometry machine. And it will tell me about each and every cell in that sample. I mean, I can decide how many cells I want to look at, but say I look at all of the cells. So it will tell me that, okay, each cell has this size and this granularity and this much of fluorescent signal, right? So it has that powerful a technique. FACS, on the other hand, is similar, okay? It's called fluorescence assisted cell sorting. This is flow cytometry plus more. Okay, so this assesses all of these properties and then it doesn't chuck the sample. What it does is it sorts the cells based on these things, these properties. So your sample is saved and sorted and saved, and you can use it for other experiments or you can continue culturing the cells. So for instance, if I'm looking again, I want to look at fluorescent protein expressing cells and I want to separate them out. So I will look at the cell, all of the cells that I have entered into the machine. I'll check, okay, are they expressing high levels of fluorescent protein or not? If they are, I will do what is called setting a gate, meaning that everything that is above, say, some X level of fluorescence, everything above that I will save and everything below that I will throw. So at the end of my experiment, I will have sorted out only the cells that are highly expressing this fluorescent protein, and I will get a pure culture from that. So here you can see you have the overall flow of how things happen in a flow cytometry. This has not included facts here. So you have this thing called sheath fluid. Sheath fluid is the liquid that moves constantly through the flow cytometry machine. And why is that important? I'll explain in a second. So you have your sample here, which is your cells in suspension. So imagine that you want to separate out or you want to look at how many cells are expressing orange and how many cells are expressing blue. So typically you will run these cells through, it sort of uh, takes the sample in through a sample injection port, and then using this sheath fluid that's flowing at a very high rate, it performs what is called hydrodynamic focusing. So I have that in more detail here. So essentially allows the cells to go one by one, then you hit it with the laser light source and you can detect these different properties. So the three main components of flow cytometry or a flow cytometer are the fluidics, the optics and detectors, and then your electronics that convert whatever has been detected as a detector into something that you can analyze, right? So the fluidics, these are what help focus the cells into a single file. So if you have a whole bunch of cells that are you're hitting with laser at the same time, you're going to get some muddled up signal, right? It's not going to be clear. So you need to look at the cells one at a time so you can assess their properties individually. Okay. So only one cell will cross a laser at one time, the laser beam at one time. So you can assess the properties one by one. So you have the sheath fluid, as I mentioned, that's flowing very fast, very rapidly, and you have the sample, okay, over here. And this sheath fluid flows in this direction and performs what is called hydrodynamic focusing. So what does that do? It presses in on your sample and creates a single cell file, okay. And that makes the cells go in one by one by one in this sort of fashion. And then as seen here, you have a laser light source that has this one bright beam of laser that's hitting the cells at this point. So at this focus point, you will have at any time, you'll have one cell. And based on the kind of emission you're getting from that sample or the kind of scatter you're getting from that sample of different kinds of light, you can determine whether it has some, what size it is, or whether it has some granularity, or whether it has the fluorescent signal of your interest. So that was about the fluidics. Now the optics. So I mentioned how we can look at size and granularity or internal complexity. So these are given by these parameters called FSC and SSC. So forward scattered light, or FSC, is proportional to the cell surface area or the size of the cell. So if this is your cell that's say moving, it's been hydrodynamically focused, and this one cell is moving through the light path, which is here where light source is written. If the light hits here and is slightly sort of deviated, then you will have this forward scatter. If the light moves within the cell and is scattered almost at 90 degrees by internal particles and granules, then you will have a side scatter, and the detector for that is somewhere here. So forward scatter is going to be around here, where you don't have a very large angle of deflection, and side scatter detections are typically around 90 degrees to the initial laser beam. So these can detect your FSC and SSC, and you can plot them. Okay, so when you're looking at your cells later, this is how it will look. This is one of the first plots you're going to see, where you have your side scatter versus forward scatter. This is someone who has run whole blood. So whole blood will have many different cells with many different sizes and granularity. So you can see how lymphocytes, or your T and B cells, they are quite small and they don't have any granules. Okay, typically they don't have any granules. So you can see that they are of small size, meaning that they are low here on the FSC axis, which is your forward scatter axis, and they have low granularity. So they're on the low side of the SSC axis as well. So they're over here. Monocytes don't have much granules, but they're bigger than lymphocytes. So they're going to be a little beyond on the FSC axis. Neutrophils, on the other hand, are gigantic. They're also very big, quite big, just like monocytes. And they also are, as you know, granulocytes. So they have high granularity. So no wonder the group of cells that are neutrophils are coming really high up, because they are high, they have high side scatter or high granularity, as well as a large size. So they're high on the FSC axis as well. Okay. So using this FSC and SSC, you can actually tell your cell population. And then suppose I only want to study lymphocytes, I'll do what is called drawing a gate. I'll draw a gate around the lymphocytes and only study that population. Then I can further see if I've done some staining for that. I can look at, okay, maybe, oh, are they activated, or are they, are they inhibitory or something. Similarly, I can do that for neutrophils. Okay. So I can take out each population and I can study it in more detail. PMTs, or photomultiplier tubes, are the most commonly used type of detector in this case. And the specificity of detection. So these PMTs, what do? They essentially multiply the signal that you're getting and give you a robust electrical output. And you have the specificity of detection is controlled by optical filters, which block certain wavelengths while transmitting some others. So if you guys know a little bit about how fluorescence works, you'll know that typically, suppose there is a dye like GFP, for instance, it will absorb light at a certain wavelength, or be, and then therefore be excited at that particular wavelength, and it will emit therefore at a higher wavelength. Okay, so it's all about, how there is an excitation of an electron, then it comes down. On the way down, there is emission, right? It's losing energy and you have an emission at a higher wavelength. So GFP, for instance, excites around 488 nanometers and it can emit around 520 nanometers. So if I want to catch this emission in the right range of emission, I want to make sure that the detector I have has a optical filter before it that can clean up the wavelengths that are coming from my sample. So I might have different kinds of emissions from my sample, but I want exactly GFP emission to be detected by my detector. So I want only that sort of general, maybe from say 500 to 540, I want that range of light only to hit my detector that's coming from the sample. Right, so I can have an optical filter that can block some other wavelengths that are say outside of that range that I mentioned, and transmit the ones I want towards the detector. Okay, and so you guys can read a little, if you're interested, about the different types of filters that exist: long pass filters, short pass filters, band pass filters. These are basically different types of filters that, and their design can determine what kind of light can enter. Like say long pass is like you will have all wavelengths above a certain thing. Short pass is all wavelengths below a certain level. Band pass is like I am defining a certain band within which I will have light going through. Like say I want only 500 to 540 nanometers to enter through this bandpass filter and hit my detector. So I'll have this filter. Dichroic mirrors, they're interesting. They are also used a lot in these microscopy and all that these days, where you can have a certain, they can be used both for excitation and emission, where you have light of a certain wavelength going through it. And when it's emitted, the wavelengths that are not needed pass through and they are ignored, while the ones that we're the wavelengths that we want pass through and are detected, while those that are not are reflected off and discarded. So using these different combinations of filters and mirrors and so on and so forth, you can actually define uh, what light is exciting your sample and what light that is being emitted is being correctly and accurately detected by your uh, detectors. Okay. And then you will get your output, which will look like this. Cell sorting, on the other hand, is a technique where, uh, as I mentioned, the sample is saved. So after you finish studying what the different properties of the cell are, and you say, okay, I only want big cells that have GFP expression, then you will deflect the cells based on that. So after this point, so here is where you have a laser that is hitting your single file of hydrodynamically focused cells. So one cell is moving here, I've hit it with laser, and here I have looked at its different properties like um, its size, its granularity, and the fluorescent markers that may or may not be present in it. Okay, so suppose I want to see again fluorescent label expressing cells only. So say one cell that has passed through, I know now that it is that it's not debris, it's not too small, and it is also expressing the fluorescent protein. So that's good. I want to save that cell. So what happens next is it passes through and it starts, then the stream will start partitioning into droplets after a point, right? Because you're dropping it from some distance, it starts partitioning into droplets. And these droplets, and you can adjust how this partitions into droplets with some different factors in the machine. And then these droplets will actually start passing through these this charged field, okay, this electric field where these plates are there to deflect this droplets. So this point where this droplet, this stream is becoming a droplet, is very important to sort of make sure that that's at the right place. So the break off point is important, and that is something that you can set up while you're setting up FACS. And then individual droplets, just like how we had individual cells that need to be interrogated by the laser, we want individual droplets to be interrogated at the electric field, to be deflected at the electric field. If we have a whole gush of cells over here, they're not going to be accurately deflected, right? So we want individual cells in droplets that are passing through this electric field and being deflected off based on whatever properties we want. So suppose I want cells that are big and are expressing my protein, they will be deflected, say, to number 4. And suppose I want cells that are big and expressing a different protein. Say these were expressing GFP, the other expressing some red fluorescent protein. I want them to deflect over here. And so they're deflecting over here. And this is waste. So everything else that is either small or not expressing either of my fluorescent protein, them I will throw away. So they will go into the waste. So now I have one tube with my GFP labeled cells and one tube with my RFP labeled cells. And these cells, we have to of course be sure to maintain sterility. After that, I can take these cells and use them for my culture. So that is FACS sorting. Okay, so I hope that is clear. And that is how it's different from flow cytometry also. So I hope that technique is clear. I'm sure you guys have heard of these techniques, maybe studied them before, and I hope this was a nice refresher on it. Okay, so now we'll go to our final few questions. All right, if anyone has any questions, please let me know or I'll proceed. Okay, so what are induced pluripotent stem cells? Can anyone tell me? Anyone? I hope you guys are awake. Is it just me in this meeting?
B.
That's correct. That's very correct. The answer is B, which is they are adult cells that were reprogrammed to a pluripotent state using the Yamanaka factors. So good job. Let's move to the next question. Which of the following are the Yamanaka factors used to generate iPSCs? Do you guys remember from what was taught? Anyone? Hmm. So the answer here is B option, OCT4 or OCT3/4. It I think has two subunits, SOX2, KLF4, and CMYC. Okay, these are useful to know. They're definitely something that you should know for um, in just generally if you're studying about stem cells. They're very important. Um, it's very important to know these factors and how they were used. Okay, and of course, in your free time, you could read about what each of these factors do in the cell, right? You guys might have heard about CMYC, right? That it is a proto-oncogene. It's a C version that is the proto-oncogene, and if it becomes oncogenic, it becomes MYC, and it can actually lead to some uncontrolled proliferation and other tumorigenic properties. So CMYC in itself is you must remember that proto-oncogenes and tumor suppressor genes all function in harmony in normal cells. But if there is some transformative event or some mutation, then it becomes harmful. So yeah, the answer here is B, OCT4 or OCT34, SOX2, KLF4, and CMYC. Okay. All right. Next question. Okay. What is the primary difference between transfection and transduction? Anyone? Anyone? So the answer here is A option. Okay. Transfection uses non-viral methods, while transduction involves viral vectors. So as I mentioned, when I was telling you about these gene delivery techniques, that transfection is when you're introducing some foreign DNA like a plasmid into some cells, and this is done without the use of viruses. Okay, so you can use electroporation, or you can use some chemical or mechanical techniques of introducing DNA into the cells. Transduction, on the other hand, is a virus based technique where you have a virus that has got the gene of interest that you want to introduce coded into its genome. So this virus will infect your cell, go inside the cell, and then it will integrate its DNA with the genome of that cell and use the host factors to produce all its genes, including that of your gene of interest. So that also introduces your DNA, but using a virus, using a biological method. So the option here, the correct one is A. Okay. And why are the other ones wrong? Transduction is temporary while transfection is always permanent. It's very incorrect. It's actually more often the other way around, where transduction typically can lead to stable line generation. You can generate a line of cells that is expressing some protein constitutively as part of its genome, while transfection typically, of course, it is possible to create stables with transfection or create um, more long-term expression with transfection using selection and all these different ways. But typically what we do in the lab is called transient transfection. So you transfect the cells, use them, you overexpress some protein and then you use them for some few days or whatever, and then you stop using them. So transfection typically is not as permanent, while transduction can be permanent based on what your experimental requirements are. C option, transfection is used only in bacterial cells while transduction is used in eukaryotic cells. That's not necessarily true, because you can also infect a bacteria with a bacteriophage, and you can transfect both bacterial cells and eukaryotic cells. So that's not true. And transduction is always performed using electroporation. Obviously, this is wrong. Transduction is using virus. Electroporation is a transfection technique. Okay. So I hope this is clear for everyone, because if you're going to work in a lab, you're going to be transfecting and transducing cells for your experiments. So it's good to know the difference between these two things. Okay. And the different methods you can use to do it. All right. Okay, next question. Which of the following parameters can be typically measured using flow cytometry? Anyone? You can unmute and say, or you can message. Okay. All right, I'm getting some answers. Everyone seems to be on the right track. The answer is A, cell size and granularity. So good job. As I mentioned, cell size can be measured using your forward scatter, granularity using side scatter. Okay. And the other things, DNA sequence variations, mitochondrial respiration rate, cell motility, adhesion. Adhesion strength you can't tell, but maybe adhesion a little bit you can tell based on the expression of adhesion molecules on the surface. But you cannot directly correlate that to adhesion strength, of course, because the proteins need to be functional for that. And you cannot sense mechanical tension or force with it. So yeah, so the answer here is A, cell size and granularity. That is something you can typically measure. And then after that, of course, once you've done that, on that cell population, you can measure your fluorescent protein expression as well. You can also measure things like, you can also check whether your cells are alive or dead. There are these live dead stains, or, you guys must have heard of, the uh, dye exclusion methods, right? Where you incubate your cells, so your live cells, on ice with this live dead dye. Okay, and this dye, if the cell is dead, what will happen, right? There are these pores in its membrane. So the dye will go inside and it will bind randomly to a lot of cytosolic proteins. And if the cell is alive, its membrane is intact, the dye will not be able to go through. So, so the cells that are dead will have a high signal of this dye, and the cells that are not will remain unstained or will remain without any signal. And so you want to check, okay, out of my, 1,000 cells, how many of them are alive? And amongst the live cells, how many of them are expressing my protein? Because it's important to look at live cells, because dead cells can take up antibodies. So you can have false positives from them. So looking at a live population when you're doing cell flow cytometry is honestly really important for your assay. So it's always good to include a live dead marker when you're performing flow cytometry experiments, particularly if you're characterizing surface levels of some protein involved in some process or the other. Okay. So yeah, the answer here after that aside is cell size and granularity. Okay. What is the function of Cas9 in CRISPR-Cas9 gene editing? So I went through this technique with you guys. I'm sure you guys know. Anyone? Someone has raised their hand. No, not anymore. Okay. Anyone? Mm, People are answering. Someone has answered E. Anyone want to try? Anyone else has any other answers about what is Cas9 protein doing in CRISPR-Cas9 based gene editing?
B.
B, right? So you're absolutely right. It does cleave the target DNA at a specific sequence. Okay, so the answer here is option B. So let's go through them one by one. So A, it's saying it acts as a guide RNA. So obviously that's not the case because Cas9 is the protein that is performing the cleaving, and the guide RNA is what is recognizing your target DNA. So the guide RNA has a complementary region that binds to your target DNA, and it also has a scaffold region that binds to Cas9. So that's what brings the Cas9 close to the target DNA, that allows the Cas9 to then cut the DNA at that sequence. Okay. And the other ones obviously are wrong. It inhibits gene expression, nope. And it inserts foreign DNA into the genome. So that's not the case at all. In the case of HDR based knock-in, Cas9 is not the one inserting the foreign DNA, right? Cas9 is just the one cutting. Then you have your template that you add in separately that has your foreign DNA. That is then added in. And of course, it's not without DNA breaks. It's after Cas9 creates a double-stranded DNA break and you have HDR based repair. Cool. So I think this is the last question. What is the role of PAM in CRISPR-Cas9 targeting? Does anyone know? Does anyone remember the importance of protospacer adjacent motif? I'm getting some varied answers here. Hmm. So does anyone else want to try? So the correct answer here is actually C option. Okay. So Cas9 needs to bind and cut the target DNA, right? And this PAM site is typically close to where this cutting will take place and close to where the guide RNA binds. And PAM is useful in the Cas9 binding near that guide RNA target region and allowing for CRISPR, the Cas9 to cut that gene. Okay. So why is A wrong? It's because it says it guides Cas9 to the correct RNA sequence, but that's not the case, right? Cas9 is typically used to target DNA. So you want your PAM site in the genome or whatever gene of interest that you are cleaving with Cas9. You want that PAM site to be near your target sequence. And your guide RNA is what will bind to the target sequence. PAM is nearby where your Cas9 binds. And that is what then goes and cuts the target DNA. So I hope that is clear. So when you are, doing CRISPR-Cas9 experiments, you want to say knock out some gene from some cell line and create a stable knocked out version of that cell line, then you want to, make sure that the PAM sequence is there, and wherever you're cutting that gene, you have a PAM sequence close to where you want that cut to take place. So PAMs are there in the genome normally also. You just need to find the right spot to cut. So there are some rules on where you want to cut in a protein, as I mentioned, right? If you are knocking out a protein, you're not actually cutting the entire protein gene and throwing it out. Protein coding gene and throwing it out, right? You are, making some breaks or some mutations in some region that makes it either not produced or non-functional. So you want to decide maybe where all you want to create the cuts so that you have a properly non-functional protein. And that you will decide based on where is a good place for, a unique place for a guide RNA to bind and whether there is a PAM sequence nearby or not. And that PAM sequence, the sequence of, for is specific to what kind of Cas enzyme you're using. So Cas9 will have some PAM sequence, Cas12 will have some PAM sequence, right? So based on what kind of system you're using, you will have to check which PAM sequence it is. Okay. So that's it. And I hope that this was educational for you guys. Yeah. Right. So if anyone has any questions, please let me know. And I think this is the last week, so I hope that you guys learned something useful from both the course as well as my problem solving sessions. Yeah. You can always reach out across the Google Groups thing that's there if you want to ask questions. And all the best for those of you who have registered for the exam. And all the best for all your assignments. Yeah. If no one has any questions, we can end the meeting then. Can you show other MCQs? Oh, yeah, sure. I can do that. Um, I will also be uploading them, of course. As you know, there is a Google, there is a Google Drive link that has been shared with you guys. This is one of them. Hey, so the person who messaged about the MCQs, this is one of them, and I think I had a few over here. So maybe you can take a look at these and see what the answer could be. And then this one, I mean, you can refer to this as well. I hope you can see the screen. So yeah, the Google Drive link, these are all available. The slides are available in PDF form for you guys. And of course, the recording is available on YouTube as well. And that link has been shared with you as well, I think. So you can check out the PDFs as well as the videos in case you've missed something or you joined late. That that should be fine. So here, yeah, 1 second. I think you are answering 3, 4, 5, 6. Okay. So I think you're telling me the answers, or oh, you want to see the next one? Okay, so this was 1, 2. The answer to this first one is 1 and 4. Okay, and the answer to this is the secondary cell wall. That's the innermost layer. Then this one, 3, 4, and 5. This was about E2 hybrid system mostly. Yeah, I hope that's clear. Yeah. So the answer to number 3 is that the E2 hybrid system is used for protein-protein interactions to study them. And in the Yeast2 hybrid system, the transcription activation will occur when you have your bait DBD and your prey AD. It will interact via bait-prey interaction. If this interaction takes place, it will bring AD and DBD close to each other, and you will have the activation domain close to the promoter, allowing it to then activate transcription of the gene. Leading, in this case, it will be the LAC-Z gene, and you will have beta galactosidase produced, which will then give a blue color upon xgal as a substrate being entered into the media. And this answer for the fifth one, which of the following is an essential negative control? Its negative control is something where you don't have transcription activation, so constitutively. So this is the one. You want to make sure that your bait DBD on its own is not causing any self-activation. So you want to see that whatever baseline there is, you want to go above that in your actual condition. Okay, so that is 3, 4, and 5. I didn't have too many other questions. Yeah, that was pretty much it. And then these. Okay, so I hope that's fine. Yeah, no problem. Good. So yeah, if anyone has any other questions or any clarifications, let me know. Everyone's all good? Okay, then thank you all for attending, and all the best for your exam and so on.